Great, thanks so much, everybody. Great, so what I wanted to do is talk to you about some of the research that we've been doing into how Oxford Nanopore sequencing can improve the manufacture of mRNA vaccines. So I'm from the BASE facility at the University of Queensland, and we're Australia's largest manufacturer of research-grade mRNAs, currently supplying to commercial, academic, and government research labs, and we're also implementing clinical manufacturing into our pipeline. So Telmo has already introduced mRNA manufacturing beautifully, so I'll just skip through this quite quickly. First of all, mRNA vaccine sequences are encoded in a plasmid backbone, and that includes the UTRs, the coding region, and the poly-A tail. This plasmid is then linearized, 3' to the poly-A tail, and it's used as a template to transcribe the mRNA, which is then purified and formulated into lipid nanoparticles. And what's really important is that at every step of mRNA manufacturing, the quality should be confirmed. For example, if the cap and the tail were missing, then that molecule of mRNA wouldn't be translated in the cell. Or if there's a mutation in the coding region, then that could impact the amino acid sequence of the protein that's translated. Or finally, contaminants such as double-stranded RNA can increase the risk of dangerous inflammatory reactions in patients. So monitoring all of these different factors in every mRNA vaccine batch is of primary importance, and that's why the United States Pharmacopoeia have come up with a draft set of guidelines about testing the quality attributes of mRNAs during their manufacture. This includes the identity, integrity, and purity of mRNAs, and this covers a raft of different methods that are complicated. They have really big machine footprints, and are quite unwieldy, leading mRNA vaccine quality testing to take about half of the time for mRNA vaccine manufacturing. So we wanted to test whether Oxford Nanopore sequencing could be useful in mRNA vaccine manufacturing, and we did a proof of principle study. And we tested exactly that. We wanted to understand, we thought there was a lot of potential for mRNA vaccines to be tested comprehensively and in a streamlined way using Oxford Nanopore sequencing, and we proved exactly that in our proof of principle demonstration. So the next step was then to validate this Oxford Nanopore mRNA identity test, which we've been doing in collaboration with Oxford Nanopore, and paving the way for the Lonza collaboration as well. And so what we wanted to do is to much more comprehensively try rolling out Oxford Nanopore sequencing throughout mRNA vaccine manufacturing. So, for example, during the Plasmid DNA release testing, also for the testing of the drug substance, the mRNA, prior to formulation, and also final drug product testing. So as part of our plasmid release testing, I just wanted to credit Sneha Lakshman Das, my colleague who's done much of the research for this part of the project. She prepared a lot of rapid nanopore sequencing libraries and sequenced them to confirm plasmid identity. What was really cool about this method is that we were also able to evaluate the contamination with supercoiled plasmids, which again produce some quite funky mRNAs, which are not good for you. So identifying them is very important. We were able to do that by looking at the read start site, and also we were able to map bulk reads against the E. coli genome and identify any sneaky E. coli reads that had snuck through the plasmid purification process. And what was pretty cool about this is that we were able to generate enough sequencing data within 2 hours of sequencing or less to provide a robust analysis. In fact, if we included additional sequencing in our data set, we saw a subtle increase in insertion and deletion error, mismatch and deletion errors, so there was no benefit at all to sequencing for the full 72 hours. Also, this was very sensitive with this amount of sequencing. We were able to detect subclonal variants at as little as 1% of the pool. We validated this by preparing some some like COVID-19 variant mRNA vaccines with about 85% similarity, and created a staggered pool. And the variant that was present at about 1% was identifiable above the background error rate or higher. So our mRNA drug substance testing now was also done by Sneha Lakshman Das and Magdalena Budzinska. So she's been helping prepare the pipelines. So how does this pipeline work? What we did was we took the mRNA product and we sequenced using RNA-004 sequencing. And then we also did base calling using Dorado with a custom model. And this custom model could identify N1-methylpseudouridine in the mRNA vaccines. This is extremely important because N1-methylpseudouridine is incorporated in mRNA vaccines, and it's an important part of their clinical efficacy. And so in this way, native mRNA fragments were able to be analysed end-to-end. The reads were mapped and then analysed to produce a comprehensive mRNA identity test report that covers various aspects of mRNA identity and integrity, which I can step you through now. So just first of all, this is the wall of data that we were able to generate for different constructs within 24 hours. We sequenced mRNAs that that used N1-methylpseudouridine in their design, and also canonical uridine, 10 vaccines in total that were sequenced in triplicate, and using different coding regions, including EGFP, ovalbumin, luciferase, mCherry, and Cas9. And so within 24 hours, we got up to 4 million reads, rather. And so we subsampled that also to 200,000 reads. And the accuracy was as follows. It was about 91 to 95% accuracy. And I believe that the base call is being refined at the moment, and those rates are improving. The highest amount of error rates were deletion error rates. Now, we were only analysing the coding region and the UTRs here because the poly-A tail had actually very high deletion error rates. Despite the fact that the poly-A tail was removed from the analysis here, there were still deletions were the highest error. I'll talk about the poly-A tails soon. We were able to confirm the sequence identity of all of the different mRNAs that we analysed. And this was very important for the reasons I went through earlier. Also, we were able to look at mRNA integrity. This was a proxy of mRNA coverage or mRNA length in our analysis, and this is extremely important to ensure mRNA stability and translation. So because long-read sequencing covers from the start to the end of the mRNA molecule, we were able to do this, and we could see that there was a bit of variation, but 89 to 95% of mRNAs were full length. So we were also able to look at the poly-A tail length. This was particularly interesting because of the very high deletion error rate. We were able to use the Dorado base caller poly-A tail length estimation. So this looks at the raw squiggle data, and it infers poly-A tail length directly from it. What was really cool was that we were able to not only confirm that the majority of the poly-A tails were of the correct length, we could also identify some subclonal deletions in a subset of the poly-A tails, and this led us to refine our manufacturing methods. So we started using a DNA-based template rather than plasmids, which has gotten around some of these truncation issues. So final product characterization. We've also developed a method to remove mRNA from its formulation. This is key to being able to analyze mRNA vaccines as a final drug product, and it's a whole slew of testing that's required by the USP. So we've developed a method that's high enough purity to sequence very, very good quality direct RNA sequencing libraries, and you're able to identify very small amounts of degradation that happened during the formulation process. And that can be great for refining the formulation process, or also for batch monitoring. Finally, what's really cool about using nanopore sequencing for mRNA vaccines is that it can analyze multivalent vaccines. So you can imagine so multivalent vaccines are increasingly being used in mRNA manufacturing to cover different subvariants or to pool together different, say, RSV, flu, for example, and COVID vaccines. And they simply can't be resolved using Sanger sequencing, right? It would give you a mess in a chromatogram. But long-read sequencing allows us to not only map all of the different reads to their respective references, it does so in a quantitative way. So what we have here is a staggered pool of different mRNA vaccines that have been pooled together between 40% and 10% of the pool, and the amount of reads that we counted, mapping to the respective genomes, gene sequences, matched the input. So that was pretty cool. So finally, our goal was to look at how Oxford Nanopore sequencing could be used during plasmid DNA release testing, drug substance testing, and final drug product testing. And we are able to say that that it does work well for the vast majority of these different applications. There are some different aspects that are still under development. The supercoiled assay is still being improved, and also 5' capping assay and double-stranded RNA assay, but we're well on our way. So thank you very much. Thanks so much.